Hi, AP Bio students. I am going to walk you through your assignment for today. We're going to be working with the classical genetics simulator, and I just want to give you an overview of how to do this and kind of how to play with it so you can complete your homework to do the challenge problem. So the first thing that you're going to do is type in cgslabs.com. You should get a site that looks like this. And you're going to go to launch CGS. It may tell you that you need to download some kind of player or something, but it should be on your computer. So just make sure you activate that. You are going to log into your account. And because you're a new student, you are going to click New Student. When you get here, the course ID is Alice. Alice. And this is going to be AP Biology, and then the year of that you're currently taking it, which is currently 2019. You're going to be working by yourself, so you're going to go to individual. You're going to type your name, and then on the next screen, it is going to give you a password. You need to, for example, it's going to give you a password. You need to record this password because it's not you. There isn't a way to retrieve it other than to contact me and retrieve it that way. So if you could write that down and then say, yes, I write down my password, you will enter into this area right here. There are 4 different populations and a challenge problem. I'm going to show you a couple of things looking at population 1 and 2. Then I'd like you to play with these a little bit, and then before you try your challenge problem. Keep in mind, as you're working on this activity, I gave you this handout in class. Your goal is to determine which phenotypes are dominant or recessive, and you want to determine if the gene is autosomal or X-linked. Okay, if you find that you have 2 autosomal genes in the trait that you're looking at, you will also need to determine if the genes are linked or unlinked. I will give you a hint. Challenge population 4, or excuse me, not challenge, population 4 has linked genes, so please make sure you look into that a little bit to see if you can figure it out. And if they are linked, you want to determine your recombinant frequency and try to figure out how far apart they would be on the same chromosome. So strategies for solving today's problems, you're going to use test crosses. You're going to use reciprocal crosses. If you need to kind of get a little more information about that, I gave you a packet last class. And then you want to perform chi-squared analysis on your crosses. The chi-squared analysis, you don't actually have to do the calculations. You just have to tell them, the program, what ratio your expected ratio is. And then it will give you all the values, and you can determine what the values mean. So as you're going through this, you need to record some information. You do not need to record information for population 1, 2, 3, and 4. You can if you want to, but you do not need to turn that in, so you don't actually have to record all this information. You need to record all this information as you're doing your crosses for your challenge population. I apologize, my son is watching cars in the background, so it got really loud all of a sudden, if you can hear that. You need to be able to have this all turned in for your challenge population. So one of the first things I suggest you do is to get yourself a nice Google Doc so you have a place to record your crosses when you get to that point. Okay. So I'm going to log into my account that I made already so I can show you how this works. Maybe.
And I'm all done with this.
Oh, I thank you, dear. You may have one more. That's fine. Okay, let me open it. Go ahead. Okay, so Wadsworth, my password. Oh, I didn't mention it. Type my password. So this is how you would use your password if you were going to come back in. That's what I actually needed, password. Okay. This is one I started earlier, and I just want to kind of show you, so because I have some things set up. So I'm gonna show you population 1. Okay, so what they originally always give you is they give you a wild population. You can't analyze the wild population and actually gain insight from it. You just have to use these organisms to figure out how the trait's inherited. When you're looking at your organisms, I usually like to organize by sex just because it's helpful for picking crosses. And then I usually like to look at the traits. This one, we're only looking at one trait. It is a little bit simpler. There may be multiple traits. So what you want to do when you're doing these crosses is to pick organisms and just drag them to your vial and do your crosses. Okay, you need one female and one male. If you're trying to cross two males together, it will tell you it won't work. So for example, I could cross this one with this one, so I could cross 2 of the same trait. Okay, I could cross 2 of this shaven trait. Okay, I could cross 2 that are different. I could cross this one with this one. Okay, you can cross and do a lot of different things. But when you're ready to do the cross, you want to press cross flies. Okay, and it'll give you a vial. In this vial, I like to also organize by sex, and I like to organize by the trait, and I like to see roughly what I'm kind of looking at, so I can see could there possibly be a ratio. If you go to analysis, it's a little small, but it'll give you the parents, it'll give you and what the offspring look like, which can be helpful. So let's go back and kind of think about things that we need to record on our sheet. So population, what are we working with right now? Population 1. You do not need to do this for every population, but you definitely should do this for the challenge population. What phenotypes do you see? So we are looking at shaven and spineless. These are bristles. Which vial? So you're going to want to make one of these tables for each vial. You do not need to show every vial that you do, but you want to show the ones that are helpful to your understanding. I would guess that you would need probably 3 or 4 in order to do this well for your populations, especially for your challenge. You're Thank you, dear. How about now? It'll be over in a couple minutes. So you want to record the vial that you were doing your crosses on. So this one would have been vial 5. I'm going to go back. I did a couple of earlier crosses. It does save all your crosses. Down here at the bottom. So okay, so if we're looking at our vials, so here is vial 1. This is one I did the other day. I look at my analysis. So I crossed number 14 with number 2, so a spineless versus a shaven, and I got this ratio of offspring. If you want to put any notes or anything down here, you can put notes down there. But you want to record this information, and I'm going to show you where I recorded it. So I made you an example. So here's my two parents. Here are the progeny. You can just copy that straight off of that analysis. And then what you need to do is you need to do some chi-square analysis. You may not know exactly what this is until you do a couple crosses, so it might be helpful to make a couple vials and then come back and do all your chi-square analysis before you kind of form all your conclusions. Oh, thank you. So if I wanted to do my stats and do my chi-squared, looking at this, I would guess this is probably about a 1-to-1 ratio, okay, which I can calculate. Okay, it'll give me these values. Okay, so I have a chi-square value of 0.3, degrees of freedom of 1, p-value of 0.5. And what you want to think about is how does that p-value relate to 0.05? If the value is greater than 0.05, then you can fail to reject your null or accept it. If your value is less than 0.05, then you have to reject your null. In this case, I'm going to fail to reject my null, okay, because I have a high p-value. If you want to look at the distribution table, you can always look at it up here. And what this means for me is, so here I have my ratio, I have all that value, I fail to reject my null. I'm going to make some conclusions about this. Okay, so I fail to reject my null, so there's no significant difference between my expected numbers and my observed numbers. So there is a 1-to-1 ratio. At this point, though, it could possibly be an autosomal or sex-linked trait, as it is possible to get a 1-to-1 ratio with autosomal inheritance and sex-linked inheritance. And I showed 2 Punnett squares that do verify that. Okay. I didn't. So I can't. I wasn't going to. Here, can you go put these back on in the other room? Yeah. I have a 2.5-year-old, clearly. I can tell this is definitely not codominance and incomplete dominance because there is not a third phenotype. I can't say at this point, though, what actually is dominant. So if I did another cross, okay, so if I go back, this doesn't give me a ton of information. It's some good starting information, but not a lot. I did an additional cross. So for this one, I crossed together shaven number 1 and spineless number 3. If you do this on yours, you probably will not get the exact same crosses, but you will get similar results. I want to look, and then here's all my progeny, which I recorded. If I look at this again, it also looks like a 1-to-1 ratio. Okay. Oops, wrong table. Okay, so here's my bio 2, also 1-to-1 ratio. I did my chi-squared analysis. I recorded it. I also failed to reject my null, so there's no significant difference between what I'm seeing in a 1-to-1 ratio. What's interesting, though, is that when I set up this cross, I picked parents that were opposites of what I had before. So if you know here, I see I have a female that's spineless and a male that's shaven. Here I have a female that's shaven and a male that's spineless. This is what we call a reciprocal cross. Because it's a reciprocal cross, if this was sex-linked, I would see something different than I saw in my previous cross. So because I don't see anything different and I'm still seeing the same exact 1-to-1 ratio, I can tell, okay, I can always organize these a little bit so you can see it. Okay, so if I look at this one, I'm still seeing about 1-to-1 here. If I look at bio 1, and I look at them as well, I'm still seeing about 1-to-1. But I'm not seeing that the males are expressing one trait only, which I would expect to see if this was sex-linked in one of those crosses. I should have seen males expressing only the reciprocal trait. And I actually didn't see that, so I can rule out sex-linked inheritance, and this must be autosomal. I still don't know what's dominant though. So what I did, I took both of those spineless parrots from the previous two crosses, and I crossed them together. When I crossed these two spineless together, let me show you what I got. This is vial 3. Organized by sex, bristles. I got this, which is still difficult to figure out. If you look at the analysis, it looks like kind of like a 3-to-1 ratio. If this is an autosomal trait and I'm crossing two of the same together and I get a 3-to-1 ratio, we can figure out exactly actually what the dominant trait is. So in this case, the dominant trait must be spineless. So I showed my Punnett square showing my 3-to-1 ratio. My final conclusion is that this is autosomal and spineless is dominant for bristles. So this is your goal, is to go through and do this. I'm going to give you a couple of little hints. Let's go back. So you do not need to record all that information. I think you should just definitely just play with your populations, but definitely record all that information for your challenge so you can figure out exactly what's going on there. If you have more than 2 traits, you should always check to see if the traits are linked. Okay, so you can go through. I will tell you now though so one of your populations was autosomal. One of these populations is going to be an incomplete codominant example. One of these populations is going to be a sex-linked example, and then one of these populations is going to be a 2-trait cross where you have to just figure out linkage. So just to give you a heads up ahead of time, that's what you're going to be seeing in these regular populations. In the challenge, though, you might see something a little bit different. So I'm going to show you a quick thing for population 4. Okay, so here's my wild population. In this one, you actually have 2 traits. So organized by sex, and then you have wing angle, and then you have wing shape. Okay, don't try to analyze the wild population because it just randomly caught flies in this case. So wing angle and wing shape. Analyze them individually. Okay, so do the exact same thing that you were doing before, but do it individually first. When you're thinking about linkage, what you want to be thinking about is how can I get across with some type of predictable ratio? So if you go to your analysis, or excuse me, your stats, I can analyze 2 at the same time, so wing shape and wing angle. Okay, and I can see what kind of ratio I would expect to get from a cross. So if I wanted to, organisms. Okay, so if I take this little guy, and cross it with this little guy, GeoVile 6. Let's go to stats. I can just figure out the ratio for this. I could just figure out the ratio for this. Or I could look at both and try to figure out what the ratio might be for both of the traits together. So I recommend that you try to get across. Okay, thanks, honey. Go find the truck and I'll fix it. I recommend that you pick a ratio that you're comfortable with. So try to do a cross that would yield a 9-to-3 to 3-to-1 ratio, or a 1-to-1 to 1-to-1 ratio, because then you can use that for doing your calculations. You can have other ratios, and that's totally fine, but I think the ones that you're most comfortable with would be easiest. So your goal for what you need to do before, I guess we have class on Monday, is to try to get that challenge population, or try to attempt that challenge population. I'm going to make this assignment due later in the week next week, so if you do need help, you can come in and get help. But I would like you to work on this for homework so that way you can get a good start on it and start playing with it and seeing what you need help with. You're welcome to work with other people in the class, but I do ask that you record your own information. Everybody's numbers are going to be different, but you might be able to use each other for support to kind of figure out what's going on. If you do need help, please come see me. This is a really kind of real life way to think about genetics. Most geneticists don't get this trait is dominant, this trait is autosomal. This is what they actually get. They get real data that they have to analyze and figure out how traits are inherited. So this is your first try at trying to analyze real data like a geneticist. Send me an email if you have any questions or come see me tomorrow. Have a good day, guys.